Welcome, everyone, to the Energy Transport Lecture for Exercise Physiology. So let's get into it. Maybe. There we go. So, first, we want to talk about your individual projects. You've all submitted your kind of goals for that. A couple of you reached out with some changes, which is great. So, I wanted to give you a little bit more information when it comes to those goals and kind of what to expect and what to do. So, with your individual projects, the directions are now up on iCollege. But for that, when it comes to writing your paper at the end of the semester, there's just kind of going over some of the things you want to keep in mind and start tracking now for that project. So the first thing is you'll get 25 points of your project from the background and introduction. So why did you pick this particular goal? What's the important for overall health and wellness? Like why is it important? And all of you should be able to connect your goal regardless of what it is to kind of the health and wellness. Um, in that kind of identifying how it relates to health and wellness, you need to use three kind of research articles, journal articles to support why that goal is good for your health and wellness. These can be very broad and how they help, but you need to find kind of the literature that backs that up and kind of talk about those and point to that in your background and introduction. And then state your goal very specifically. Use a SMART goal that's measurable with an outcome. Some of the goals that you guys submitted in your projects weren't quite smart, but I could see what's there. So just make sure you're finding those and what those look like. After that, you will kind of go into a method and your plan, like what are you planning on doing? What were your methods to reach your goal? And so include the frequency, duration, the time, like how it was progressing through the semester, what your time frame was. So really put that down on paper and figure that out now. It'd be a lot easier to track as well. Be very specific on how you are going to support yourself on like test weeks and things like that throughout the semester, but also like how does your program actually match with research on how we're supposed to program or do behavior changes. Um, Describe what your measure, of course. Like, are you going to use a stopwatch to measure like a time component or a Garmin watch for that? Or are you going to use kind of weights or a scale and what scale that is? And so just be specific where it's really clear what you're doing and how you're going to do that with both your main outcome and kind of some of your secondary outcomes that you're looking at. Then you'll have 25 points for results and outcomes. So you're going to measure your progress over time. How often are you doing that? Include your baseline assessment. So if you haven't done that, you really need to go ahead and get your baseline assessments done. And then your final assessment, like what was that goal? Include graphs to help depict your results. These are always helpful. Again, if you're tracking weight over time or 5K time over a certain set of months, you can see how that's progressed. Make sure your graphs are labeled, have captions, describe if you're missing data, if something occurred maybe a certain week, or if you didn't stick to your plan, or whatever it may have been, you're going to want to put that in there as well. Again, both main and secondary outcomes. And then you've got 25 points for your discussion. So briefly re-summarize your goal, discuss if you did or didn't meet your goal, why or why not, like what were some of those issues. You're not graded on the fact that you didn't meet your goal, so don't lie. Just be honest about it. You're learning through this project and what you're doing. Interpret and explain your results, like what were the trends did you see? When were you successful? When were you not? Um, how did that influence your health? How you felt? Things like that. And then what could you have done differently and give recommendations to other people with similar goals. And so trying to wrap that up all in that paper that you'll write. So just be thinking about these things now and start tracking things down and make it a lot easier. Additionally, you'll get 10 points for your grammar and writing quality. Proofread your paper, have someone else proofread your paper, things like that. Look for errors, make sure it's clear and concise, make sure that anyone that picked it up could understand what was going on. And then your transitions, just try to make those as smooth as possible. Just again, working on some of those writing skills as well that you'll need as you move into the PT world. You have to be able to communicate well, both written and verbally. So that's kind of your project. You guys can talk to me in class or email me if you have questions as we move forward. So one thing I want to, before we jump into the next topic, I do want to jump back quickly to last week's lecture where we talked about the glycemic index and glycemic load. we talked about it. I had to jump back and forth, got it wrong. The higher the number on glycemic index, the more it's going to spike your blood sugar. Where the lower numbers, it's going to more slowly have a gradual rise in your blood sugar. And then glycemic load is looking at kind of the rate of carbohydrates and how they're going to be processed and the amount of carbohydrates in them. So glycemic load is a more accurate picture of food's impact on your blood sugar. And so it's just trying to make sure we get that in there. Glycemic load is looking at that difference between total carbs and fiber content and how those balance, and just gives that better idea of food impacts. And so just put that up there just for us to discuss it again, but we're not in class today. So for example, on this, you could use something like watermelon has a high glycemic index, but a low glycemic load. Okay, when you look at this chart, its carbohydrates raise blood sugar really quickly. But typically, if you think about watermelon, it's not a lot in there. It's mostly water. It's very high water content. So the glycemic load isn't that heavy, sheerly because of that quantity of glucose in there. And so just kind of keep that in mind. So with that, let's jump into kind of what we're going to focus on today, and that's kind of energy transport, energy production, really, when it comes to the body. And so here's some learning objectives. You can read through those, but I know it's there, and maybe some of you it'll help, but we'll just kind of jump on. You can pause and read those if you want. You also have access to this lecture on iCollege. So when it comes to energy, we've got to remember that energy, when we talk about it in really any part of this world, the first law of thermodynamics, I know we're going all the way back to that, and that is that energy is never created or destroyed. Energy only transforms from one form to another and is never really depleted. Energy is always there. A lot of times it's transferred into heat, and then that heat sometimes maybe can be recaptured and transform that energy back into something more usable, depending on what we're looking at. And so it's really important for us to remember the human body doesn't produce or consume energy, it just transforms energy. Plants are the same way. We sometimes say they produce energy from the sun, but really they're just changing that radiant energy into a different type of energy. And so when it comes to the human body, the energy that we kind of use to transform into usable sources is from food. We simply break food down that has stored energy in it, and we break those bonds down to give us something that we can actually utilize for energy. So the entire goal of our digestive system is to break food molecular bonds down into smaller pieces that then we'll convert through other ways that we're going to talk about into energy that then our body can directly use for energy. And again, just changing that form. And so the only usable form of energy in the entire body is what's called adenosine triphosphate, or ADP. Sorry, ATP. Adenosine triphosphate, ATP, is the only usable source of energy in the body. If we can't make ATP, then we don't have a usable form of energy. Doesn't matter what we consume. If we can't make it into ATP, then we can't have our daily life functions go on. And so that's really important. So here's the chemistry structure of that. You can see the adenosine group, there's a ribosome group, and then there's 3 phosphates. The energy that is holding those phosphates, the bonds that are holding those phosphates together, are very high energy bonds. And so breaking those apart kind of releases energy for us to use for our daily processes in the body. So ATP will eventually lose a phosphate group. It'll be broken down into ADP, so adenosine diphosphate, and it breaks this one bond over here, gets rid of that phosphate group, releases energy for us to use. And so that's really what's going on when we're kind of creating energy, quote unquote, is we're breaking down ATP into ADP and losing that phosphate group. And then the body works really hard to convert that ADP back into ATP so we can use that for energy. So when we look at that adenosine triphosphate, just like what I said here, ATP plus a little bit of water, H2O, transforms into ADP, adenosine diphosphate, some inorganic phosphate plus energy. And so that's what we're going to be able to utilize in the body. So, um, we can talk about some of the amounts. Like if you like the chemistry aspect, energy is about 7.3 kcals per mole. But you may be more like thinking about it like, what do we use ATP for? Like when we get this energy, what does it do for the body? Well, it does things like active transporting calcium in our muscles. It will do sodium potassium pumps. It helps with our cross bridge cycles in our muscles. So literally helps with muscle contraction. We need ATP for that. The creation of DNA and RNA, the synthesis of that, and the synthesis of any protein in the body. Our neurotransmitters use it. So any kind of electrical signal being transmitted throughout the body is going to need ATP. And so it's really needed for everything in our body. Because our body works on kind of gradients. We create a gradient of high concentration to low concentration. And anytime we're trying to force something into a high concentration, it's going to take energy to push it there. We're going to have to push things there, hold them there, and then release them when the time is right, and then recapture that. And so we're always using energy for that. And that's going to come in the form of breaking down ATP into ADP. So, couple things to note with ATP is at rest, a person stores about 80 to 100 grams of ATP. So we could pull that out. If we pulled all the ATP out of your body, it would weigh somewhere between 80 and 100 grams. And so you may say, well, that seems like a good bit. But in all honesty, that's only about a few seconds worth of life. If you stop making ATP right now, you would only live for about 3 to 4 more seconds, probably before your ATP is run out. You have no energy, and therefore you would die. So, with that being said, we store very, very little amounts of it in all honesty. And so, every second, a person is making about 2.5 moles worth of ATP. And that equates to, if we look at it on molecule scales, 1.44 times 10 to the 10th power. So a whole lot of ATP molecules are produced every single second. And if we're physically active, when we're physically active, we need a lot more energy, right? Our muscles are moving. We need a lot more energy because we're using more ATP. So we have to produce more ATP. So that number can skyrocket once we start exercising, especially at higher intensities. And so if we don't store very much of it and we need this much of it at rest and a whole lot more when we exercise, well, where does it come from? And so that's really where the rest of our lecture is going to process today is where does that ATP production come from? And it comes from three main sources. And we're going to talk about those sources in order from kind of fastest producing, the one that produces the ATP the fastest, to the one that produces it the slowest. Now realize that's a relative scale. All of these things will happen in less than a blink of an eye. But relative to each other, the first one produces it the fastest, the last one produces it the slowest. And then remember, there's always a trade off with speed. Think about it. If we run at the fastest that we possibly can, we can't run for very long. But if we slow that pace down into something slower, we can run for a very long time. So as we go through these systems, we're not only going to look at which one creates the most ATP the quickest in a given time frame, we're also going to see which one lasts the longest. So the one that makes it the slowest is going to be able to last a lot longer. And so there's trade-offs, right? There's always a trade-off when it comes to things in the human body. So the first system we have is creatine phosphate system. The creatine phosphate system is a one-step reaction to get ATP. It only takes one step. And so creatine phosphate provides energy. Typically, if we think about the hardest activity you can do for about 15 seconds. So when you initially start exercise, creatine phosphate system is going to be the main system involved as the others kind of shift and ramp up and start making ATP because they're a little slower, they take a little bit more time. But the creatine phosphate system's only good for about 10 to 15 seconds in all-out activity. So if you can do it for more than 10 to 15 seconds, we are starting to talk about depleting that creatine phosphate system or other systems playing a significant role in it. If it's less than that, creatine phosphate may be the only system in play of ATP production in that activity when it comes to vigorous exercise. And so creatine phosphate, what happens in that process is we take creatine phosphate and an ADP, so adenosine diphosphate, and we, through a one-step direct phosphorylation process, we create ATP and creatine. So really remember, it's in the name. We looked at that chemical structure of being phosphate bonds. Basically what happens is that creatine phosphate, the phosphate is donated to ATP or to ADP and becomes ATP, and we're left with just simply creatine. And so that's how that process works. The enzyme that shifts creatine phosphate into ATP and creatine is called creatine kinase. Okay, and so it's a one-step process, and it goes back and forth. Creatine phosphate, or sorry, creatine kinase can make that reaction go in either direction, like these arrows show. So in times when we need a lot of ATP, the reaction will force creatine phosphate into creatine ATP. In places like when we're resting right now and we don't have this high demand for ATP, it'll shift back, and what you'll have is ADPs and creatine phosphate. And so just remember that reaction is reversible. It goes both directions. And so how does it know when to do one over the other? Well, it all comes to levels. So when we have high levels of ATP, we shift towards ATP production. So if there's a lot of ADP available, that reaction will shift towards creating creatine and ATP over creatine phosphate and ADP. When we have high levels of ATP, so there's lots of it, and so we're not depleting those energy sources, the reaction goes the other way, and we stop creating ATP through creatine phosphate. The good news with all of this is that our muscle cell stores about 4 to 6 times more creatine phosphate than we do ATP. And so that means we can go for longer than those couple seconds, like what we were talking about with just ATP stores. But this 4 to 6 times more gets depleted quickly, right? If we're only talking ATP stores lasting, 2, 3, 4 seconds, well, if we multiply that by 5 or 6, what you'll see is you'll get around 12 to 15 seconds worth of energy. So it does last longer, but it does deplete quickly. Even though it's a multiplier, it's just not that big of a time difference. Realize that creatine phosphate is produced heavily in the kidneys and liver. If you've ever seen supplements with like L-arginine in it or glycine, this is the system they're kind of focusing on. And so supplements can be helpful in this, but you got to remember it's not going to be helpful, but in the right circumstances. So explosive power and strength and short bouts of activity, because this system lasts for such a slow period of time or a low period of time, it's not going to help you with a marathon per se. And don't forget, we will have waste products here of creatine. And so if we have too much of that, maybe the body doesn't produce it its own, and it can have issues over time. So in addition to creatine phosphate, again, here on the right-hand side, you can see the kind of the chemical structure of taking that creatine phosphate, ADP, going into creatine and ATP, again, just losing that phosphate, as seen on the group. This is an anaerobic system, so it doesn't need oxygen is what that means. You can not breathe, and this system will still run. You can hold your breath for 15 seconds and still live, and so this is part of that process. This creatine phosphate process also happens in the cytosol of the muscle, or the muscle cytoplasm, maybe you've heard about. So just in kind of the fluid inside of a cell. It doesn't have a specific organelle that it forms in. It's just kind of in this reaction is occurring very quickly inside the cytoplasm or cytosol. And so just kind of keep those things in mind. So the big thing when people hear creatine phosphate energy system is they're like, creatine supplementation. That's where this is connected. And yes, that is the side the idea of this. And so typically those will contain something like a creatine monohydrate is what you'll see a lot. And so this will convert in the body to a creatine phosphate. And so they're looking for things like this. It is relatively safe. There are few side effects from it. Some of those may be nausea, GI issues for a lot of people. If you have some kidney or liver problems, it can play an issue, but again, for most people, it's relatively safe. It can help with explosive power and strength, but realize that half the population is what they call a non-responder to creatine, meaning that It doesn't matter if they take creatine or not, it's not going to help them. So half the population really doesn't respond super well to creatine supplementation. But the other half that does can see benefits if they're doing multiple bouts of explosive activity. Otherwise, they may not. But it can help with making your muscles look a little bigger because you retain water from it. It can help with some hydration things. And so the recommended dosage if you get into creatine supplementation is about 5 grams a day, give or take. And if you're interested in learning some more, there's a short snippet of a podcast that you can watch, and it talks about that and some people who are definitely for it and giving you that recommendation. And so always good to kind of look at your information and what it's really good for when you're taking supplements and spending money. So that's our creatine phosphate system. And so that's the quickest system, but as you can see, wears out really quick. It's only good for about 15 seconds of all-out activity. And so if we're just going for a run as fast as we can, let's say we're getting chased by T-Rex, getting you up and out of your chair is your ATP stores, and then phosphocreatine system is going to get you basically out the door, right? Think about wherever you're sitting, getting up out of a chair, running from a T-Rex in your room. It'll get you to out the door, but we can run further than that. So the system that kind of kicks in after that becomes the main energy producer of ATP at that point is a system called glycolysis. And so if you look at the word glycolysis, lysis at the end of a word means to cut. And glyco is simply short for glucose or table sugar. And so what happens here is we just simply are cutting table sugar up in order to make ATP. And so this is when we talk about consuming carbohydrates and breaking down into glucose, glycolysis is what is going to convert that glucose into our usable form of energy, ATP. And so this is where all of that starts, is through glycolysis. Glycogen as well enters this system. Glycogen is also enters glycolysis and is broken down into ATP. This system also occurs in the cytoplasm of the cell. So any cell that we're looking at, it's in the cytoplasm of that cell, or the cytosol, depending on kind of what term you're familiar with. And again, this system is anaerobic, meaning it doesn't require oxygen to operate. So do keep those in mind. So if we go down to some of the chemistry and looking at this particular process, we'll get into this picture to the right. Basically, what I like you to see is the cytosol. You have glycolysis occurring, and then eventually we'll move into this mitochondria, and we'll talk about the other systems. But right now, glycolysis is anaerobic, doesn't require O2. And so what happens with glucose is it's a 6-carbon molecule. And if we take glucose and what we call NADs, which are kind of hydrogen carriers or electron carriers, we take 2 inorganic phosphates. So remember, ATP needs that phosphate group that we talked about, so we're going to need some of those, and 2 ADPs, right? We can kind of see where this process is headed. If we run glucose and all these things through glycolysis, you'll get 2 pyruvates, which are 3 carbon molecules. You'll get 2 ATPs, you'll get 2 NADHs, and you'll get 2 waters. And so if you do all this math here, basically what you see is we make some water, ATPs and NADHs from what we have, and we convert this glucose from a 6-carbon molecule down into 2 3-carbon molecules. So it literally gets cut in half. It makes 2 things. And so our net reaction, what we truly get when we look at it with the glucose, is 2 pyruvates, 2 ATPs, so we make 2 energy molecules, and 2 NADHs. And so keep in mind these NADHs. When we get to the very end of our lecture, we'll be talking about what happens to these, but these will be used to make more energy at the end of the day. So if we continue with glycolysis, where oxidative phosphorylation was a one-step process and happened very quickly, glycolysis still happens quicker than our last energy system, but not as quickly as the creatine phosphate system, because it's a 10-step process. And so it's 10 reactions long. We take glucose and get it to pyruvate, from glucose 6-phosphate to fructose 6-phosphate to fructose 1,6-bisphosphate to dihydroxyl acetone phosphate. And as you can see, we continue to move things on from glyceraldehyde 3-phosphate, 1,3-bis phosphoglycerate, 3-phosphoglycerate, 2-phosphoglycerate, phosphoenol pyruvate, and then pyruvate. I know, hearing me read a lot of words that I am terrible at pronouncing. That being said, you can see the numbers a lot of times. We are just shifting the chemical structure of a lot of this, and then eventually we are kind of transforming that into some different things. So with glycolysis, it's really important that we note 4 key enzymes. So each one of these processes back here, there's an enzyme involved for that conversion. Glucose to glucose 6-phosphate, there's an enzyme. That enzyme is hectokinase. Okay, it's the first step in our process. And what happens here is it converts glucose 6 to glucose 6-phosphate. That's step 1, just like what I just mentioned here, right? And so you have this one, and then you have number 2 right here, phosphofructokinase, converts fructose 6-phosphate into fructose 1,6-bisphosphate. And so why do I point these 2 out over others? Well, these first 2 steps, or these first 2 these 2 steps that we're talking about here, because they're not perfectly our first 2 steps, right? It gets us all the way to step 4, but these 2 right here, the reason we talk about them is these 2 processes require us to input ATP in order for them to function. So they actually take energy for them to actually occur. So at hexokinase and phosphofructose kinase, we're going to use 2 ATP for these processes to occur. And so that's really important for us to remember that it actually takes ATP to run glycolysis. So we do invest some energy to get some energy out. It's also important to notice to note that phosphofructose kinase is our rate limiting enzyme. This is the one step in the whole process that is rate limited, meaning this is where things will back up. If we don't have enough phosphofructose kinase, which is our enzyme that we're going to have the least amount of out of all of these, this whole process will stop or come to a crawl. And so that's really important to know. We then get on to phosphoglycerate kinase, which converts 1,3-bis-phosphoglycerate into 3-phosphoglycerate, and then pyruvate kinase, which converts phosphoenol pyruvate to pyruvate. These 2 steps, the reason we point them out is we produce ATP at these 2 steps. So at each of these 2 steps, you will create ATP. So you may be thinking, well, we take one glucose molecule and we put 2 ATP in and we only get 2 ATP out. How does that efficient? How does that help us? Well, between these 2 steps here, number 2 and 3, what happens is our glucose molecule goes from a 6-carbon molecule into a 3-carbon molecule. And so we take 2 ATP in, but when we split that molecule into 2 separate ones, it'll basically run through these 2 processes twice. And so what'll happen is you invest 2 ATP to get 4 out of it. And so again, summary here, this glycolysis process occurs in the cytosol of the muscle. It's the starting product of glucose or glycogen. It uses ATP, and our end product is 2 pyruvates and 4 ATP. And so this over here is that chemical structure and goes through all the steps. But the important one is to see up here with hexokinase, that ADP, wrong side, glucose with hexokinase here, it takes ATP to make that first step happen. We go a couple more, and right down here we see it takes another ATP in order to convert to fructose 1,6-bisphosphate. And then here with fructose bisphosphate aldolase, so an aldolase is a splitter, we split that 6-carbon molecule into 2 identical 3-carbon molecules. And then they continue on and we make some NADHs right here. And then we create a ATP. And then at the very end again, pyruvate kinase will create another ATP. And so this process happens twice as indicated by the arrows. And so we'll make 4 total ATP with an investment of 2. So at the very end, again, we have this pyruvate. So what happens to our pyruvate is very important for us to kind of note and talk about for a little bit. And so pyruvate has 2 different pathways depending on the presence of oxygen. If oxygen is not present, so we're in an anaerobic condition, the pyruvate will convert into the lactate cycle, the lactate cycle. If it's aerobic condition, pyruvate will transform into the Krebs cycle. So let's talk about the anaerobic condition first. So lactic acid, pyruvate will be converted into lactate, which eventually will convert into lactic acid, and then it'll we can talk about the Cori cycle at the same time. So in the absence of oxygen, pyruvate converts to lactate through lactate dehydrogenase, pyruvate to lactate, as you can see, and you're going to take 2 NADHs, and you're going to make 2 NADs, and you're basically going to give hydrogens over to this lactate, which you can kind of see coming over here. That being said, pyruvate to lactate dehydrogenase, there should be an arrow pointing the other way because this is a reversible step. So pyruvate to lactate can bounce back and forth. It's a two-way reaction, just like creatine phosphate is. So when this process occurs and we make this lactate, the NADs are basically the hydrogens are given up, the NADs are freed up, and so this allows additional glycolysis to occur when oxygen is not available. So it's all about running glycolysis for longer. The lactate can go all the way back to the liver to be converted back into pyruvate, but it can also happen in the muscle itself. Like heart muscle loves lactate to convert that back to pyruvate and use that for energy. And so it's happening in all of your muscles as well. And then we can even look at pyruvate converts to glucose via gluconeogenesis in the liver as well. So if we do get lactate back to liver and it's converted to pyruvate, that pyruvate can be made back into glucose to continue glucose sparing initiatives. So if you look at that kind of quickly, the lactate converts pyruvate back at the liver and kidneys, and then it undergoes gluconeogenesis. These are the steps for that process. You can kind of see how it's going to move through. It's going to donate and go through some processes. The one thing to note here is that converting pyruvate back to glucose requires 6 ATP. And so you can see that this is a very cost inefficient process. It's not what the body prefers. We like to be efficient because at the end of the day, you are going to use 6 ATP to create that new glucose molecule. It'll go through glycolysis and take another 2 ATP. So the total investment here is 8 ATP to get only 4 ATP back through that process. And so that's really a negative result and not what the body prefers, but it is something we can do like in long activities. The body's more likely to do this, or if we're in a food deprived state, so we're kind of in starvation, the body will do this more readily just to make sure we have enough glucose for the brain and other life essential functions to continue to operate. So one thing to note is with this process, remember it's anaerobic when oxygen is not available, so glycolysis is running. We continue to run and build pyruvate, which will turn into lactate in an oxygen deficit environment, in an anaerobic environment. And so the problem with lactate is when we have lactate anywhere, it quickly and very easily dissociates into lactic acid. And so you should really think lactic acids usually aren't great in the body, right, except in certain places. And so we talk about hydrogen ions there. And so what happens with lactic acid is it creates an acidic environment. And so think about acids, they kind of break things down. They make things not work. Think about pouring acid on your skin. It burns, it starts to degrade. And so in an acidic environment anywhere in the body, things start to not work as well. And so in, let's say, a muscle fiber, if we have a buildup of lactate, which converts to lactic acid, it becomes an acidic environment. And so that acidity starts to inhibit glycolysis. It starts to break down the enzyme. It interferes with calcium binding sites on troponin, so kind of how our muscles move. And so too high of levels actually shut down muscle function and energy production. And so that's going to interfere with our movement, and that's all about trying to make sure that we're not creating so much energy through glycolysis that we have major long-term damage. It shuts everything down before that occurs. And then once the activity stops, the body will clear that lactate, that lactic acid. It'll turn it back into pyruvate, and we'll use it eventually. But it does cause issues for kind of continuing exercise per se. And so we do want to make sure that we kind of look at that. We never want to build up a ton of lactic acid. And the reason that's important when it comes to exercise is we can measure that buildup of lactic acid or lactate in the body. And if it ever starts to get too high, we know that glycolysis will stop, and eventually our muscles will stop moving as well through that calcium inhibition. And so it's important for us to know in activity something called lactate threshold. And this is simply the point in which blood lactate levels start to increase almost exponentially, where the body can't clear the lactate levels. And so lactic acid builds up, which will start to slow down energy production and decrease our muscle capabilities. And so if we look here, this graph kind of shows lactate threshold. It is simply looking at running treadmill speeds in kilometers per hour, and we measure blood lactate. So we all have lactate in our blood all the time. We can measure it right now. Do a simple finger stick. We just prick the finger, take a drop of blood, and with an analyzer we can see that. And then when we start exercising, that amount will increase, and it'll slowly increase with speed. If you notice here, we're slowly increasing lactate levels with speed, but very little. Our body's easily able to handle this amount that we're rising by. But as we increase that speed more and more, what happens is we hit lactate threshold, and this is the point where lactate goes from increasing gradually to increasing rapidly, almost an exponential rate with increased speed. And so what happens at this lactate threshold point, this is the point where if we go above this lactate threshold point, lactic acid starts building up, and we can't maintain this kind of activity above lactate threshold for very long. Our muscles and our energy system just can't keep up and keep things cleared, and so they'll shut down. And so really, lactate threshold is a representation of anaerobic threshold. Above this, we're anaerobic, and we can't keep anaerobic activities up very long. And so the whole goal is to stay below lactic threshold for endurance athletes. Endurance athletes are always trying to get up to lactate threshold as close as they can, but never go over it, because as soon as they go over it, they start to feel bad, their muscles start to burn, and they start to slow down. Where if we can stay just below it, they can maintain as high of an output as possible for a very, very long time. And so there are ways to improve lactate threshold. We can either do it one of two ways: by decreasing the amount of lactate being produced, or by increasing the rate of lactate clearance. More aerobic training really close to lactate threshold will kind of reduce lactate production. And if we do things like energy or interval training, where we go above lactate threshold and then back down, above lactate threshold and back down, that will help us with the clearance process. And so both of those will help with us being able to maintain a higher kind of intensity level for a longer period of time without having this buildup of lactate, which is not great for our activity. So with that being said, we talked about pyruvate and what will happen to pyruvate in anaerobic conditions. It'll go into the lactate cycle, which is a reversible thing. In aerobic conditions where oxygen is present, pyruvate will enter the Krebs cycle. And so we will take pyruvate, we'll enter the Krebs cycle. The Krebs cycle happens in the mitochondria, or the powerhouse of the cell. Maybe you've heard it to that. So every cell has mitochondria, and inside of that is where a majority of energy is made, and this is where our aerobic systems occur. So that being said, pyruvate is converted pyruvate is converted into acetyl-CoA, which will be in the mitochondria and then enter the Krebs cycle. This is really the first step of the Krebs cycle, is acetyl-CoA entering the Krebs cycle to make citrate. The pyruvate to acetyl-CoA transformation that occurs is one way, meaning that this is an irreversible step. Once pyruvate is converted to acetyl-CoA, it never goes back. And so this is a committed step of, once it hits there, it will go through Krebs cycle or the aerobic cycle. Additionally, as we're moving through the Krebs cycle, we'll create some NADHs and FADHs, which will enter our electron transport chain, which will help us to produce around 36 ATP. So we have this kind of 36 to 38. This is for one molecule of glucose, we will produce around 36 net ATP. When we subtract out our 2 ATP we had to pay in order for these systems to run at the beginning. So let's jump into kind of that first forming of acetyl-CoA. So we have pyruvate here, and if we take the pyruvate with an NAD8 or an NAD, and we add a coenzyme A, what will happen here is through pyruvate dehydrogenase, we will create an acetyl-CoA. So you can kind of see that transformation. So 2 pyruvate, because 1 glucose molecule makes 2 pyruvate, you'll get 2 acetyl-CoAs, 2 NADHs, and 2 CO2s. So carbon dioxides that we'll breathe out. And so again, that enzyme is pyruvate dehydrogenase, and that is an irreversible step. Once we go into acetyl-CoA, we can't go back. So that acetyl-CoA will then enter the Krebs cycle. So it's way up here in the green. This is a cycle. It's called that because it kind of loops back into itself, and it keeps going as long as we have the things to feed into it, like waters and NADs and acetyl-CoAs, and we have all the appropriate enzymes. This cycle will just continue and keep producing ATP and NADHs and FADHs. So acetyl-CoA will come in and with citrate synthase create citrate. That will then move through this cycle, and you can see at different stages. Actinase will produce a water molecule, and we'll go to cis-actinate, and then we'll take some water in, even though we just got rid of some, but it's all about transforming that molecule, and actinase, and we'll get isocitrate, and then isocitrate dehydrogenase will produce an NAD, right, H. We'll take an NAD, produce an NADH, and make some CO2, some carbon dioxide, and we'll get alpha-ketoglutarate. Alpha-ketoglutarate dehydrogenase will take a coenzyme A, another one of those, NADH to make NADH, an NAD to make an NADH. We'll produce some more CO2, and we'll get succinylcholine-CoA. And then it has a synthase. You can kind of see how these are labeled pretty well. And you'll get succinate. In this process right here, which is really cool, we'll actually produce an ATP for every time we go through this cycle. So for a glucose molecule, we'll produce 2 ATP directly from Krebs cycle because of having 2 pyruvates come from that. Again, this will move through. We'll get FAD to FADH. So these are similar to NADHs, but a little bit different, and we'll show how those are different here in a minute. And then eventually this works from fumarate to maltate to oxaloacetate, which is kind of the, if we want to call it, the ending phase of the Krebs cycle, because then we just restart by adding acetyl-CoA back into it to make citrate. And along the way, if you're a chemistry person, you can see how this kind of changes through chemistry and goes from a 5-carbon molecule down to a 4-carbon molecule, okay? And so these are those processes. One thing to note is isocitrate dehydrogenase is our rate-limiting enzyme here, just like we had for glycolysis. There is a rate-limiting enzyme, and so this will be what kind of slows everything down. So just kind of keep that in mind as we go through. So things we've already talked about is input to acetyl-CoAs. What we get if you kind of track everything and kind of count it up 2 times around the cycle is you get 4 CO2s, 4 carbon dioxides that we'll breathe out, 6 NADHs, 2 FADHs, and we'll produce 2 ATPs. And so that kind of ends that. And so how do we get that 36 number, right? Well, it's all about what do we do with these NADs and FADHs that we have produced. And so let's talk about how we take these NADHs and FADHs and convert them into more ATP. And this is through the ETC, or the electron transport chain. The electron transport chain occurs in the mitochondria as well, just like the Krebs cycle. The Krebs cycle occurs in basically the very center of the mitochondria, where the electron transport chain occurs what they call in the inner membrane space of the mitochondria. So the mitochondria has 2 membranes. So think about it, 2 cell walls. It's got the outside one, which most people think about, and then it's got an inside one. And then you've got the center. The center of that mitochondria is where Krebs cycle occurs. And then along the inner membrane, that you can't see, it's kind of tucked away in there, are these proton pumps, which help us make more ATP and is the electron transport chain. And what happens is between the inner and outer membrane, we have this inner membrane space. And this is really important for us to be able to build up a concentration of hydrogen ions that will help us to make that ATP. And so within the inner membrane, there are all these kind of complexes and proton pumps that'll help move those hydrogens. And eventually, once we pump enough of them and there's a concentration gradient, we'll be able to make ATP through the flow of those electrons from the inner membrane space back into the center of the mitochondria. And so this is a good look at it on this picture at the bottom. Ooh, too many. You can, if we look at this being the outer membrane, this kind of line here is the inner membrane. This matrix is where the Krebs cycle is happening, and this inner membrane space is this buildup of hydrogen ions through these complexes, pumping them in to eventually come through and make ATP at this kind of proton pump here. And so a hydride is basically is equal to hydrogens plus 2 electrons. So think about hydrogens in this case every time we talk about them being electron carriers. They basically just carrying electrons over, which is basically putting a potential energy in that inner membrane space that ATP synthase will take those electrons and be able to make ATP with. So kind of forces that inorganic phosphate onto the ADP to kind of have a high energy bond that we can break to utilize for energy. So again, all that being said, these complexes are pumping electrons into this high energy state, into the inner membrane space. They don't want to hang out here. They're all too alike. There's a high concentration here, so they just want to get out. And so eventually they will get out and make ATP. So when we look at that electron transport chain, look, we have complex 1, we've got 2, 3, 4. And if we look here, complex 1 takes NADHs, converts them into NADs, and pumps hydrogen ions through, again, carrying that electron. Complex 2 takes FADHs and breaks them down into FADs, so kind of freeing up some more hydrogen and hydrogen ions. Complex 3 then we'll pump those hydrogen ions, as well as complex 4. Again, all about creating that gradient up at the top. And then complex 4 is where we kind of utilize some oxygen. And what will happen in complex 4, down here if we look at complex 4, these electrons that we kind of helped move these hydrogen ions over, we'll take and combine with an oxygen atom along with some other loose hydrogens and make H2O. This is why we breathe. This is why we call it the aerobic system, because it takes oxygen in order for the system to keep going. If this system stops, Krebs cycle stops, and then that means that glycolysis runs and it will convert into lactate instead of acetyl-CoA, making it an anaerobic system. When oxygen is present, we're breathing right, we're getting oxygen to the cells. This system runs and we become an aerobic system into the Krebs cycle and electron transport chain. Remember, this system is our last system producing energy. It is the slowest relatively to produce energy, but it produces a lot of energy with one cycle, and it is able to go for a very long time, being a little slower, but it does require oxygen to be present, which is why we breathe and continue long-term sports. This O2 becomes an electron acceptor at the very end, and the entire reason we have a respiratory system and we breathe in air and breathe it out, CO2, but the whole reason we have that respiratory system is to get rid of some CO2 and to breathe oxygen in to get it to the muscle, to come to complex 4 and make some water. If this process worked differently and didn't need O2, we'd have no reason to breathe in oxygen whatsoever. And so it's just kind of a cool thing to look at. And then the very end, we have the ATP synthase that produces ATP, which again, we saw over here. So that's kind of what our electron transport chain is. It's all about getting that concentration gradient of hydrogens, which will then eventually come to the ATP synthase. This is kind of what this looks like if we look at it at a molecular scale. This is kind of that membrane we're looking at, the inner membrane space, the matrix down here. It's got all this kind of, but basically to sum up, ATP synthase takes those hydrogen concentration. They're in a very high in the inner membrane space. They diffuse into the inner membrane through this ATP synthase, and it basically causes this to rotate. This kind of rotation that occurs here because of that electrical charge and potential energy, and it forces an inorganic phosphate to bind to an ADP, and we create ATP. And that's really what we're occurring and how we're getting that energy created. And so with the electron transport chain, we can kind of start to calculate how many ATPs. And so an NAD will give us 3 hydrogens. An FAD will yield 2 hydrogens into that inner membrane space. And for every hydrogen that moves through the ATP synthase pump, we'll produce 1 ATP. And so we can start to do the math. In the Krebs cycle, we produced 6 NADHs, which is the equivalent to each one of those making 3 hydrogens, or 1 ATP per NADH. Sorry, they'll make 3 hydrogens, which each of those hydrogen converts into an ATP. So 6 times 3 is 18. We produce 2 FADHs per glucose molecule, remember, so it's 2 turns of that Krebs cycle. Those produce 2 hydrogens, which are equivalent to 2 ATP, so 2 times 2 is 4, plus the 2 ATP it generates on its own, so 18 plus 4 plus 2 equals 24. Transition, there were, remember, 2 NADHs created when we went from pyruvate to acetyl-CoA. So again, we'll get another 6 ATP here. And then glycolysis, remember, produced 2 NADHs as well. And so you'll get another 6 ATP, as well as the 2 ATP that were created at the end of that. And so if you add all those numbers up, you'll get 36 ATP made per glucose molecule. And so that's what we kind of look at here. The aerobic respiration of glucose, a 6 carbon molecule plus 6 oxygen atoms, will give us 6 CO2s, 6 H2Os, and 36 to 38 ATP. So 36 if we subtract those 2 ATP we invested at the initial part of glycolysis, 38 if you just count the gross. So just kind of keep that in mind. And so we get CO2 from Krebs cycle, H2Os come from our electron transport chain, and ATP come from both. So that's aerobic respiration, Krebs cycle. Remember, our limiting factors to this is oxygen consumption, and not just what we can breathe in, but what we can actually get to the cell to make water. If we run out of oxygen, that electron transport chain stops really working, which means Krebs cycle stops working, which means glycolysis converts pyruvate into lactate over acetyl-CoA. NADHs and FADHs, right? How our ability to transform these into pushing those hydrogens across the inner membrane space. And of course, our enzymes. There are lots and lots of enzymes in this whole process in order to get that conversion. And so if any one of those is lacking, it'll slow down, or if we're missing some of those, so it's really important for us to have a well-balanced diet to make those, make sure our body's working properly. But again, isocitrate synthase is our rate limiting, and so it's typically what will be the bottleneck when it comes to aerobic respiration. So realize when it comes to the regulators of ATP production, what determines the process glucose will go through is a few things like we've mentioned through this whole lecture, which is exercise duration and intensity. The higher the duration, the higher the intensity and the shorter the duration, the more likely we're going to be using glycolysis, and we're going to be using it anaerobically. The longer that duration, and especially if we're below lactate threshold, where we're not building up the lactic acid that's going to cause glycolysis to kind of slow down and stop, and energy, or our muscles to kind of stop reacting as well too, means we're going to go more aerobic respiration. And so part of that comes to our presence of oxygen. And really the biggest thing that kind of predicates all this is the presence of ATP and ADP. If we have a lot of ATP available, so that means we're not using that ATP, we're going to inhibit Krebs cycle because we don't need to make energy. We're not needing to make it because we're not utilizing the ATP we have. If we have an abundance of ADP, meaning we have a lot of places where we can attach phosphates, which means we're using up a lot of our ATP, because the byproduct of using ATP is ADP, then we're going to turn Krebs cycle on. And so it's really kind of that's really what's dictating it. When we use all the energy and it sees that we have a lack of it because we have too many ADPs, we'll ramp up energy production. And then when the ATPs start to build up and we're not utilizing, our energy productions will kind of slow down. And so with that, that is the end of our slideshow. And so went through it a little quick. It's not as long as you would have in class, a little less examples. If you have questions, please let me know. But this should get us there, and then we'll have lecture the next time I see you. We'll jump into the next thing. Let me know if you have any questions, and I hope you guys have a wonderful afternoon.